Hello everyone, welcome back to the channel. Today I am going to discuss about introduction to cell. Have you ever wondered what makes up your body or any living thing around you? The answer is something incredibly small but extremely powerful. That is the cell. The cell is the smallest and the basic unit of life. Every living organism from tiny bacteria to humans is made up of cells. In fact, this idea comes from the cell theory which tells us all living things are made up of cells. Cells are the basic unit of life and all cells come from pre-existing cells. So the cells are actually mainly two types are the first one is prokaryotic and the second one is eukaryotic cells. So the prokaryotic cells actually they are very simple and without a nucleus. And in the case of eukaryotic cells, they are complex and with a nucleus. Each cell performs essential functions like energy production, growth, and reproduction, keeping us alive every second. In this video, we will break down the structure, types and functions of cells in a simple and easy way. So in this slide, you can see the definition of cell. The cell is the smallest structure and functional unit of life. So it is a very simple definition. And a cell is the smallest unit of body that can carry out all the life process like growth, reproduction and metabolism. And there are two types. So the difference also you can see here. So the prokaryotic has no nucleus, no membrane bound organelles, no true nucleus, and it is unicellular and the size will be 0.1 to 5 meter and it has cell wall and it has asexual reproduction. And the case of eukaryotic, the eukaryotic cell contains membrane bound organelles, contain true nucleus and they are uni or either the multicellular. The size is little bit bigger than the prokaryotic, 10 to 100 micrometer, and they have either asexual and sexual reproduction. Now let's understand the two main types of cells, eukaryotic and prokaryotic. And the first we can see that is eukaryotic cells. The cells are more complex and have a well-defined nucleus enclosed by a membrane inside. And you can see the structure like the mitochondria which produces energy, and here you can see ribosomes which help in protein synthesis. There is also a nucleolus inside the nucleus which plays a role in ribosome formation. So the mitochondria you can see here which is developing or helps for the synthesis of energy, or it will produce ATP, and these are the ribosomes which help for the synthesis of proteins. And here you can see the nucleolus and the nuclear membrane, and there we can see the smooth endoplasmic reticulum and the rough endoplasmic reticulum. The eukaryotic cell structure I will explain again one more time. So the second one is prokaryotic cell. So we have the prokaryotic cell here, and the cells are similar and do not have a true nucleus. Instead, their genetic material is present in a region called the nucleoid. They may have a capsule for protection and a flagellum for movement. Unlike eukaryotic cells, they look membrane-bound organelles. So here we can see the prokaryotic cell and the nucleoid is here and ribosome is present here for the protein synthesis, and the cell wall in some eukaryotic you can see, and unlike the eukaryotic, you can see the prokaryotic cell has a flagellum. So these are the characteristics, features of eukaryotic and prokaryotic. Simply you can remember that eukaryotic cells are complex with a nucleus and prokaryotes are simple without the nucleus. Now let's closer look at the structure of a eukaryotic cell. At the center, we have the nucleus here, and which is enclosed by a nuclear membrane. So here you can see the nucleus, and it is covered by a membrane called that is nuclear membrane. And here you can see the nucleolus. Nucleolus is here, and again you can see the mitochondria here. The mitochondria are known as the powerhouse of the cell. They produce energy required for all the functions. And surrounding to the nucleus, you can see the cytoplasm. This is the cytoplasm. This yellow color is showing the cytoplasm where all the cellular activities takes place. And the endoplasmic reticulum has two types: rough endoplasmic reticulum and smooth endoplasmic reticulum, where the ribosome, this red dots are indicating the ribosomes. Where the ribosomes are absent, they are the smooth endoplasmic reticulum. So you can say like rough endoplasmic reticulum, which helps in protein synthesis, and smooth endoplasmic reticulum, which produces lipids. The next one is a Golgi apparatus, which modifies and packages protein for transport. And the next one you can see that is lysosome. The lysosome helps in digestion and removal of waste materials, while peroxisomes detoxify harmful substances. And the cytoskeleton maintains the shape of the cell and helps in movement. All of this is enclosed within the plasma membrane, which controls what enter and leaves the cells. So you might heard about endocytosis and exocytosis. If anything entered inside the cell, then you can call that is endocytosis. And if anything exiting from the cell, then you can call it as the exocytosis. And let me explain again. Here you can see the nucleus, and the nucleus has nuclear envelope, and the chromatin are present, which is associated proteins. And nucleolus are the condensed region where ribosomes are formed, and peroxisome, which metabolizes the waste, and endoplasmic reticulum smooth and even the rough. Cytoplasm is here, and the vacuole, these are the suicidal bags. And cytoskeleton consists of microtubules, centrosome, intermediate filaments, and microfilaments, which give structure and shape to the cell, same like our human skeletal system does. This slide has what is cytology. Cytology is the scientific study of cells. So here we can see again the cell structure, and the cell mainly divided into three types: plasma membrane, cytoplasm, and nucleus. So here you can see the cytoplasm and the membrane, the plasma membrane. You can see these are the membrane which is protecting our cell. So it's a barrier you can say, and which is covering your entire cell. And inside you can see this yellow color. These are the cytoplasm. The cytoplasm where all the metabolic or all the functions which carry out here, and the nucleus is here, and all the nuclear organelles you can see: the mitochondria, the centrosome, microtubules, mitochondria, and Golgi apparatus, vacuole, peroxisome. So each and every organelles you can see in this slide. Let's begin with the plasma membrane. Plasma membrane separating the cell from the outside environment. It comprises specific embedded proteins which help in exchange of substances in and out of the cell. The main function of the cell wall is to provide structural strength and support, and also provide a semi-permeable surface for molecules to pass in and out of the cell. So I have already told you, like there is our cell membrane, the cell membrane which is separating the cell from outside environment, and their functions are protecting the cell, which controlling what enters and leaves. That means exocytosis and endocytosis. Endocytosis means when the nutrients entering into the cell, and when the waste leaves from the cell, that you can call it as exocytosis. And the plasma membrane acts as like a security gate, and here you can see the entire structure of the plasma membrane or the cell membrane. So now we can see the close look of the plasma membrane, and it is mainly made up of phospholipid bilayer. Each phospholipid has a hydrophilic, that means a water loving head, and a hydrophobic, which you can call like a tail, water repelling. So the hydrophilic heads face outward towards the water, while the hydrophobic tails face inward, forming a stable double layer. And it is embedded within this layer are proteins. Some are integral proteins which pass through the membrane and help in transport. Other are the peripheral proteins which are attached to the surface. You can also see cholesterol molecules which help maintain the flexibility and stability of the membrane. The sugar chains attached to the proteins and lipids play a role in cell recognition and communication. The sugar chains attached to the proteins and lipids play a role in cell recognition and communication. This structure is also called the fluid mosaic model because the membrane is flexible and contains a mixture of different components. The cell membrane controls what enters and leaves the cell, maintaining the integral balance of the cell. So even here you can see everything, the cholesterol part you can see, and the integral proteins are here, and even the channels are here which allows the molecule or the ligand molecule to enter, or the receptors will be here, like even G protein coupled receptor, ligand gated channels. So this is the structure of the outward phase and even the inner phase of the plasma membrane. So after the cell membrane, you can see the cytoplasm, and it is a gelatinous liquid that fills the inside of the cell, and it composed of the components of the cytoplasm are water, salts and various organic molecules. The cytoskeleton is present there, so which consist of microfilaments, microtubules and fibers to provide perfect shape to the cell, and it is anchoring the organelles, so all the organelles are in a position because of the I mean like a cytoskeleton, and which is stimulating the cell movement. So here we can see the functions of the cytoplasm, so which is acting as a cushion and giving support and protecting the cell organelles like a nucleus, mitochondria, ribosomes and etc. other organelles, and it is providing a medium where important chemical processes like a glycolysis, protein synthesis which takes place. And cytoplasm helps in the movement of substances like nutrients, enzymes and waste materials within the cell, and it maintains the cell's shape and keeping it firm by filling the cell's interior, so which stores the water, salt, sugar and other essential chemicals temporarily, and plays in cell growth by allowing the cells to stretch and divide. So these are the functions of cytoplasm. Mainly all the cell organelles are in the cytoplasm, and it has the cytoskeleton also. So cytoskeleton which is providing the shape and structure to the cell, and even it giving the anchor to the all the organelles, and their functions you can see, you can read the functions of cytoplasm. It's very clearly symbol. So the next one is cytoskeleton. It is a network of tiny protein fibers present inside the cytoplasm, and it is made up of microfilaments, intermediate filaments and microtubules. So you can think the cytoskeletons are same like our human skeleton or skeleton. So first it provides shape and support, helping the cell maintain its structure. Second, it helps in movement, not just of the whole cell, but also movement of organelles inside the cell. For example, white blood cells can crawl using this system. Third, it holds organelles in place, keeping everything properly organized, and it plays a important role during the cell division, forming spindle fibers that separate the chromosomes, and of all the functions you can see here. And here we can see the microfilaments and microfilaments here, and the intermediate filament and the microtubules, and even you can see different types of parts like alpha tubulin and the beta tubulin, and the end part you can see here, and Yeah, this is how they are closely. Like you can see the structure here, and which keeping all the cell organelles in a place and the movement of the organelles. So like a fluid mosaic model also happen because of the structure of the cytoskeleton. So this is all about the cytoskeleton. Hope it's clear. So here we can see the all the cell organelles, the nucleus, mitochondria, ribosomes, endoplasmic reticulum. There are two types of endoplasmic reticulum: smooth endoplasmic reticulum and the rough endoplasmic reticulum. And the Golgi apparatus, lysosomes, peroxisomes, and centrosomes. These are the major organelles. Not only those organelles, there are many types of organelles are there. The major cell organelles are thus, and here we can see the nucleus, and the nucleus is here, and endoplasmic reticulum. This is the smooth endoplasmic reticulum because there are no presence of ribosomes. And here you can see the rough endoplasmic reticulum which has the projection, so that means they are having the ribosomes. And here you can see the Golgi apparatus structure and the mitochondria where the ATP production occurs, and the centrosome, lysosome, and the ribosome. The ribosome structure you can see here where the our transcription and translation happen. So this is all about the cell organelles, which this all the cell organelles are specialized structures, so which are performing their specific functions which are needed for the cells proper functioning and the survival. And here we can see the nucleus, and the nucleus structure, and the nucleus encompasses the nuclear membrane, nucleoplasm, chromosomes, and the nucleolus. And the nuclear membrane, it is a double layered structure and which is surrounded the nucleus. It is outer layer is connected to the endoplasmic reticulum. So the endoplasmic reticulum, there are two types are there, and it is connected with the nucleus. Like the cell membrane has a phospholipid bilayer, right? So same like here, the nuclear membrane also has bilayer, and which maintain the shape of the nucleus and regulating the movement of molecules through nuclear pores. And here we can see the nuclear pores. Through this nuclear pores only, the proteins or like essential things will end comes out of it, and which allowing the pores are allowing the large molecules like proteins and RNA between the nucleus and cytoplasm. So through these pores the proteins or either the RNA comes out and it will enters into the rough endoplasmic reticulum or the smooth endoplasmic reticulum. So in the structure here we can see the nuclear pore, chromatin, and the nucleolus in the middle, and nucleoplasm same like the cytoplasm, and nuclear envelope, which these are the structure of the nucleus. So when you explain about the nucleus in the paper, you can write the first, before that you need to draw the diagram and explain all the label. You need to label, that is very important, and write the functions of the major functions are like it's allowing the proteins or the large molecules to enter or comes out of the nucleus. So these are the functions and about the nucleus. So in the nucleus structure, the first one we have here, that is nucleoplasm and nucleolus, and the last one is chromatin. So the nucleoplasm means also you can call it as the karyoplasm. It is same like a gel-like structure of our cytoplasm, and it is a, or you can say again it's a gel-like a substance inside the nucleus that protecting its contents and helps maintain its shape. It also acts as a medium for transporting important materials like enzymes and nucleotides. So the second one is the nucleolus. It is a dense membraneless like structure and responsible for making ribosomes. And interestingly, it disappears during cell division and reappears afterwards. So in the cell division lectures, you will study detail about this change. And finally, the last one, that is chromatin. So it is made up of DNA and proteins, and it contains the genetic information of the cell. And the chromatin has two types. It exists in two forms: heterochromatin and or the inactive condensed form, or euchromatin, and it is less condensed and active in transcription. So the heterochromatin tightly packed and inactive, and the case of euchromatin loosely packed and active in gene expression. So the nucleus is not just a storage center, it actively controls and regulates all the cellular functions that you need to remember very carefully. And the next part is mitochondria. Most probably we heard many times the mitochondria is the powerhouse of the cell. So of course it also has double membrane organelles that is producing energy in the form of ATP through cellular respiration. Yeah, and it has outer membrane. Outer membrane is smooth and permeable to small molecules, and the inner membrane has a folds inside that you can call cristae to increase surface area for ATP production. And inside you can see the cristae. This cristae which allowing the formation of more ATP, and there you can see the matrix inside, and it is a fluid filled space which contains enzymes, DNA and the ribosomes. And you need to remember one more thing: the mitochondria owns their own DNA and ribosomes which can produce some of their own proteins and enzymes. So this is the structure of mitochondria. For 2 marks or the 5 marks, anyone can ask the mitochondria. So there you need to explain this with the structure. Structure is very important in the case of human anatomy and physiology. Whenever you are writing exam, you need to draw this diagram with the proper labeling. It's very important. And here you can see the mitochondria. So what are the functions of mitochondria? So first of all, it helps for producing ATP through aerobic respiration. So that's why we are calling it as the powerhouse of the cell. So now you can answer, why does the mitochondria called as the powerhouse of the cell? Because it produces the ATP through aerobic respiration. And it helps for breaking down carbohydrates and fatty acids for energy. The second function of mitochondria. And it regulates calcium ion concentration in cell, generating heat in brown fat cell, that you can call it as thermogenesis. Thermo means temperature, genesis means formation, for the generation of heat, and controls programmed cell death by releasing cytochrome C. Not only in HAP, pathophysiology you have in you have in cell death or cell injury. So for the apoptosis process, cytochrome C is releasing from the mitochondria. Once the cytochrome C released from the mitochondria, then only the normal cellular death will start. If the cytochrome C is unprogrammadly released, that we can call it the cell death or the necrosis. Okay, and helps in synthesis of some amino acids and steroids. So these are the major functions of mitochondria, very important. So we have completed the nucleus, right? So the nucleus connected with the endoplasmic reticulum, and the endoplasmic reticulum is a network of membranes in the cytoplasm acting as a transport system and helps in synthesis of proteins and lipids. The rough endoplasmic reticulum structure we can see, and this is a smooth endoplasmic reticulum. So rough endoplasmic reticulum which help for the synthesis of proteins, and in the case of smooth endoplasmic reticulum helps for the synthesis of lipids and steroids. So I have divided into two types here. You can see RER or the SER, smooth endoplasmic reticulum and rough endoplasmic reticulum. Rough endoplasmic reticulum covered with ribosomes. So that is why we are calling it as the rough endoplasmic reticulum, because of rough appearance of it, and helps mainly for synthesis of proteins and transporting the proteins to to the Golgi apparatus. First we have studied that the nucleus is there and nucleus is connected with the endoplasmic reticulum, and here you can see the rough endoplasmic reticulum is connected with the Golgi bodies. So once the protein synthesized, it will enters into the Golgi apparatus, and smooth endoplasmic reticulum lacks ribosome. The major difference between the smooth and rough endoplasmic reticulum, that is, the rough endoplasmic reticulum has ribosome and the smooth endoplasmic reticulum has lacks of ribosomes. So it has a smooth texture. And the function: synthesis of lipids and steroid, detoxifying the drugs and poisons, and storing calcium ion, especially in the muscle cell. So this all are the functions and the difference between the endoplasmic reticulum of both. And here we can see the structure of the endoplasmic reticulum. The nucleus is here and it is connected with through the pores. The proteins and will enters inside ribosome forms and it enters into the rough endoplasmic reticulum. And in the rough endoplasmic reticulum, ribosome helps for the formation of proteins from the transcription and translation process. In exam, you can draw this diagram. This is very drawing friendly. This structure you can draw in the case of exam. So next, the ribosome. Ribosomes are very small and non-membranous organelles, and it is made up of RNA and proteins. So the major site for the protein synthesis in the cell, that is ribosomes, and the ribosomal RNA and the proteins found free in cytoplasm or attached to rough endoplasmic reticulum, which is composed of two units, the large subunit and the small subunit. So might you heard about the 60s and the 30s, so that's about the subunits. And the major functions of the ribosomes are synthesis of proteins by translation process of messenger RNA from the nucleus, and free ribosomes make proteins for use inside the cell. And of course, the ribosomes are present not only on the surface of endoplasmic reticulum, even in the cytoplasm also you can see the ribosomes. They are the free ribosome which help for the protein to make the proteins for the cell use itself. And the ribosomes on the rough endoplasmic making the proteins for exporting or the membrane insertion. So the major difference you can see here, free ribosomes are for the proteins formation for the cell use, and here you can see the structure of the ribosome. And here you can see how the transcription occurs. In the nucleus, there you can see the DNA. From the DNA to messenger RNA, formation of a messenger RNA from the DNA, that you can call the transcription process, and the protein formation with the help of ribosome from the messenger RNA, that process you can call it as the translation process. And here you can see the nucleotides. With the help of nucleotides, accordingly the amino acids will be formed. And the next cell organelle, that is Golgi apparatus or Golgi body. So the Golgi body actually made up of four to eight flat cup-shaped sacs that you can call as the cisternae. So in some organism it is very less, and in the case of humans there are 40 to 100 sacs of the cisternae are present. And I have told you earlier, the nucleus and the endoplasmic reticulum are connected. So the proteins will receive into the endoplasmic reticulum, and the endoplasmic reticulum and the Golgi apparatus are connected. So the protein enter into the endoplasmic reticulum to it enters to the Golgi body. So Golgi body help for modification of the proteins. For example, it adds sugars, phosphate groups, etc. So it packs inside the Golgi body and it will forms the vesicles. There will be formation of vesicles, and that vesicles will send to the different places like lysosomes, plasma membrane, and outside of the cell, that you can call it as the exocytosis. Also, it helps for the transportation of lipids, formation of lysosomes, and making glycolipids and sphingomyelin. So these are the functions and the process of the Golgi apparatus. In the next slide, you can see very detailed. So in this diagram, you can see that the proteins are formed from the endoplasmic reticulum, and the protein is entering into the Golgi body. So you can see the protein here, and it is entering into the Golgi apparatus, and you can see the vesicles there. So it will enter into the Golgi bodies, and here you can see the structure, the cisternae you can see, and there will be formation of the vesicle like secretory vesicle, and slowly it is moving towards the cellular membrane, so it will be enter outside. So this is the exocytosis process. And the next cell membrane, that is lysosomes. So the lysosomes are having sphere shaped sacs, and which is filled with the hydrolytic enzyme, and it has the capability to breaking down of many types of biomolecules. So of course these are the enzymes. The pH level of the lumen lies between 4.5 to 5, and making it quite acidic, and this is somewhat comparable to acidic environment of the stomach. So this is very important. And the breaking down biological polymers, lysosomes are involved in various other cellular processes. So that are also another function of the lysosome, and it is degrading discharged materials. So energy metabolism, cell signaling, and restoration of the plasma membrane, it is very important. The size of the lysosomes varies, with the largest ones measuring more than 1.2 micrometer. Through they typically range from 0.1 micrometer to 0.6 micrometer. So this is all about the lysosome. So lysosome, you need to remember, they are the suicidal sacs, or it helps for breaking down of the biomolecules, and the lumen pH is varies. So 4.5 to 5 is the pH value, and it is quite acidic. So this is all about the lysosome. Lysosome also very important, you need to remember. And here you can see the structure of the lysosome. So the lysosome has the hydrolytic enzymes inside, and the lumen you can see. So the lysosomes works as the waste discarding structure. So it is working like by processing undesirable materials and degrading them, both from the exterior of the cell and waste constituents inside the cell. And sometimes digestive enzymes may end up damaging the lysosomes so themselves, and this can cause the cell to die. So this is also another one type of mechanism of cell injury or the cell death, and it is termed as autolysis. Autolysis means the cell itself dying because of the lysosome present in the own body, and the disintegration of the cell by destruction of its cell, that you can call it as autolysis. It's very important. So if anyone asks what is autolysis, then you can say that the cell itself destructing, so that you can call autolysis. And so this, because of this reason, we are calling it as the suicidal bags, so which help for the degradation of the waste material which are present inside the cell. So suicidal bags. So the next cell organelle, that is peroxisomes. So the peroxisome is a small membrane bound organelles which is found in mostly in the eukaryotic cell, especially abundant in liver and kidney cells. So you need to remember that the peroxisomes abundantly present in the liver and the kidney cells, and it is playing a major role in the case of detoxification and metabolic reactions, so which is involving in with the oxidation oxygen. So here also, and you can read that, and it is surrounded by single membrane and which contains enzymes like oxidases and catalyzers. So in the structure itself, you can see the membrane is there and the enzymes are present, and here you can see the crystalline core, and here we go the transport proteins which allowing the movement of the molecules to in and out. And one more thing you need to remember, the peroxisome doesn't have DNA or ribosome, and it is mainly depending on nucleus for the proteins. And the major functions of the peroxisomes are which help for the breaking down of harmful substances, for example alcohol, drugs, toxins, and especially it is very available abundantly present in the liver cells, right? Of course, it major help will be for the detoxification, and it produces the hydrogen peroxide using oxidases enzyme. So it helps for the breaking down using catalase enzyme, so which preventing the cell damage. And even fatty acid breakdown also by like a beta oxidation of very long chain fatty acids will convert them into the shorter chains for the mitochondria. That also major function of the peroxisomes. Lipid synthesis also happen here, which help for the synthesis of phospholipids and plasma allogens, and that also very important in the case of brain and myelin. And there are very like this are the this are all about the peroxisomes. Peroxisomes is very important. So here we go. The centrosome is the last cell organelle of this lecture. So the centrosomes actually it's a small organelle found near the nucleus in the animal cells, and it is acting as a main microtubule organizing center. So or else you can call it as the MDOC. And the structure of the centrosomes consists of two cylindrical structures called centrioles. So like here you can see the structure of the centrosome. So the centrioles are the cylindrical structures which is present, and it is arranged at right angles to each other, and each centriole is made up of microtubules arranged in 9x3 patterns. So here you can see that, and the major functions of the centrosome you can see here. So like why it is centrosome, why the centrosome is very important. So it helps for the formation of spindle fibers during cell division, and it organizes the microtubules which maintain cell shape. It ensuring proper separation of chromosomes during mitosis. So in the case of cell division, the centrosome is very important there. So before the cell divides, the centrosome duplicates, then the two centrosomes move to opposite poles of the cell and form spindle fibers that pull the chromosomes apart. So in the cell division, you will study detail about the centrosome and the functions all you can see here, and this is all about the centrosome. Hope the cell and the cell organelles have clear, and yeah, that's it. So let me revise the topics which we have covered in this series. So you need to remember about the cell. The cell is the basic unit of life, and the cells are two types, prokaryotic and eukaryotic. Here we have explained only about the eukaryotic. So the eukaryotic cells are contain membrane bound organelles, and it has a cell membrane first. Cell membrane is a selective barrier made up of phospholipid bilayer and which controlling the entry and the exit of substances. The second we have studied cytoplasm. Cytoplasm is gel-like a matrix where all the metabolic reactions occurs. And next the nucleus inside, and it control the center of the cell and contains DNA and regulates all the activities. And the mitochondria is the powerhouse of the cell which produces the ATP through cellular respiration. And endoplasm endoplasmic reticulum, and it has two types are the rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum help for the synthesis of proteins, and smooth endoplasmic reticulum help for the synthesis of proteins, and it helps for the detoxification of the drugs also. And Golgi bodies which modifying, packaging and secreting the proteins. And lysosomes are the suicidal bags of the cell which are digesting the waste and damaged organelles. The peroxisomes which help for the detoxification of harmful substances and break down all the fatty acids. And centrosome will be form spindle fibers and helps in the cell division. And one more thing I forgot to tell here, that is chloroplast. It is present only in the case of plant. So that is the major site for the photosynthesis containing chlorophyll. That's why the plants are having green color. And one more part of the cell is there, that is vacuole. So vacuole helps for the storage of water, nutrients and waste largely in the plant cell. So this is all present in the case of plants.